Hello everyone, so this video is going to be covering the pentose phosphate pathway. Now the pentose phosphate pathway is going to be dealing with glucose, specifically glucose 6-phosphate, but instead of putting it through glycolysis and breaking it down into pyruvate, we're just going to be sort of recombining it and reconfiguring the molecule in order to get a very different result. So it's important when going through the pentose phosphate pathway to sort of keep track of the number of carbon atoms, because they're not going to change much, but the way they change is very important. The whole point of doing the pentose phosphate pathway is that we're trying to regenerate NADPH. And we're doing that in the first couple steps of this pathway. So we're going to be taking NADP minus and turning it into NADPH. And the first major steps of the pathway that do this are going to be this very first step, and then we have a reconfiguration step, and then this third step here as well. Now these first three steps are called the oxidative portion of the pentose phosphate pathway. So I'm just going to write in where we get our NADPHs, and it's going to be NADP minus for both. And so let's talk about then, once I get this in, hopefully you can read that. Once I get this in, okay, so what are we doing in terms of products? Well, we're taking a glucose, and we're going to be turning it into a 6-phosphogluconate. And we do this in two steps. The first step utilizing a dehydrogenase so that we regenerate our NADPH here. And thankfully the enzymes are pretty mercifully named. So this one is just going to be glucose 6-phosphate dehydrogenase, which I'm just going to abbreviate as DHASE. Now this next enzyme, since this is just a reconfiguration step, there's not going to be any extra products involved, but this next enzyme is simply called a glucolactonase. Now going from 6-phosphogluconate, we're going to again have a dehydrogenase step, and that's going to give us ribulose 5-phosphate. And there's also going to be a decarboxylation step. So we're losing a carbon here. So while we retained our 6 carbons here, we're only going to have 5 carbons here. And this is achieved by the enzyme, also mercifully named. It's just going to be 6-phosphogluconate dehydrogenase. So I'm just going to write this up here to keep things clean. Now this makes up the oxidative portion of our pentose phosphate pathway, and we can actually abbreviate this whole pathway as follows. We can just write the glucose 6-phosphate, which is going to go G6P, and we're going to add in our 2 NADP minuses, and then this we're going to turn into 1 ribulose 5-phosphate plus the 2 NADPHs we get from it. And finally, of course, the 1 carbon dioxide molecule that pumped off from the glucose. So that's the overall oxidative portion of the pathway. Now, what about this sort of more complicated part down here? Well, we simply take our ribulose, and depending on what enzyme we can use, we can turn it into 2 different structures. First, we can turn it into a xylulose. And xylulose has multiple roles, but it's going to be a xylulose 5-phosphate. Or we can take it and turn it into ribose. Note the difference there, ribulose or ribose 5-phosphate. And so the 2 enzymes that are involved with this are going to be an epimerase for the xylulose and an isomerase for the ribose. So for our number 4 enzyme here, it's going to be pentose epimerase. And for our ribose, it's going to be phosphopentose isomerase. Now here's where things get sort of interesting, because we can only use one of these enzymes on one of these molecules, so we actually need multiple ribuloses to complete this non-oxidative portion of the pathway. So what we can do then is if we have two ribuloses, we can combine xylulose and ribose together with the help of TTP, and we can get a 7-carbon molecule, a acetoheptulose. Since there's 10 carbon molecules total here, we'll have 3 carbons left over, so we just get sort of a generic C3 here. And so, as I said, these are 5-carbon molecules for both of these. And then what enzyme is responsible for this combination? Well, this is going to be a transketolase for this 6th enzyme. And this final 7th one, I'll just write in to complete our thing, is going to be a transaldolase. And the difference between these two is mainly the fact a transketolase is going to be responsible for 2 carbon molecule chain fragments, while transaldolase is going to be responsible for rearranging 3 carbon molecule chains. So what can we do with this acetoheptulose and this generic C3? We can actually take this C3 and reconfigure it using our transaldolase, and we can change it into a C4 instead. And also we can get a final product here, which is going to be a fructose 6-phosphate. And we can take this fructose then, and we can reconfigure it back into glucose. We can run it through the pentose phosphate pathway again, or we can put it over into glycolysis. We can do a lot of things with the fructose. So what about the C4 fragment? Well, we can take it and get a 3rd ribulose to change into xylulose, and using a transketolase, we can get another fructose 6-phosphate, so that we now have 2 different means of getting fructose out of this pathway. And I'm going to go outside the box here. But we can also get, since this is going to be 9 carbons total, and this fructose is only 6 of them, we're also going to end up with the glyceraldehyde 3-phosphate, which you may remember from glycolysis. So we can take this, we can turn this into 2,3-BPG and actually affect our oxygen affinity within our cells, or we can simply plug it back into glycolysis and use it as a precursor to get more pyruvate. So we can take this non-oxidative portion and we can put this into a summary as well. So for this non-oxidative portion, we're going to need 3 total ribuloses, ribulose 5-phosphate again. And we're going to need 3 of them so we can get our 3 different main products here. So we need 3 ribuloses, and we can bind these in different ways to get 2 xylulose 5-phosphate, and also 1 ribose 5-phosphate. And then we can recombine these 3 molecules to get ourselves 2 fructose 6-phosphates, which is going to be 12 carbon atoms total, and 1 glyceraldehyde 3-phosphate, which I'll abbreviate as GA3P. So that's the summary of the non-oxidative portion of the pentose phosphate pathway.